Can everybody hear me okay? I hope the partners can hear me okay. If there are questions that come in, please, I'm going to want to start by talking a little bit about the journey and a little bit about the company, but I want to have a discussion about, ways academia can form new companies and how they can be populated and things like that, because we worked a lot on that in the Idea to IPO class, which is the introductory class where we kick off the technology entrepreneurship series of the Collette School of Business, and we've got some good models that we think might be applicable here and might be applicable at several of the partner networks schools as well. So just a little bit about me. I'm originally from Princeton, New Jersey. I guess when I was young, I kind of was exposed to a lot of biopharma without knowing it, because now I'm interacting with them, for real now. Now that we have a biotechnology company. I actually have been around the country a little bit, got my BS degree in biology at Grinnell College in Iowa, did my PhD at Dartmouth Medical School. That's where I met my significant other. She's here as the Associate Dean of Graduate and Postdoctoral Affairs at UAB still. We were both recruited down to UAB 20 years ago, actually, this past December holiday period. Did my postdoc at Johns Hopkins, and it was from Johns Hopkins where we were both recruited down to UAB. So I've been studying human cell physiology and pathophysiology since I first started learning patch clamp electrophysiology as a graduate student at Dartmouth. Always have wanted to compare diseased human cells to normal human cells to see what is different, what is wrong, what is dysfunctional in the diseased human cells versus the normal cells. I've always had this kind of issue, as it were, that you're going to develop these therapeutics for human beings anyway, so why not do the studies on human cells, both normal and diseased primary cultures and cell lines. And so I bring that up because that's the core principle in our biotechnology company as well, and I'll tell you a little bit more of that going forward. I was a tenured faculty member in physiology and biophysics that's now part of cell biology here at UAB in the Joint Health Sciences from 2000 to 2007. But at that inflection point, 2006 to 2007, I began thinking about a paradigm called humanized drug discovery or human cell-based drug discovery, and began thinking about that first as an academic collaboration with the Discovery unit here, then as a scientific core where we focused on human cell-based types of drug discovery bioassays, and that eventually morphed into the company called Discovery Biomed. I actually attended the Idea to IPO class and Technology Venture Business Planning class as a faculty member while I was forming this company. And then a few years later was recruited in to direct and teach the first course, Idea to IPO, and then be involved in some of the other entrepreneurship classes. So that tells you that I tell the students on the first day that I was sitting where you were sitting, so many years ago. And that's really how valuable the technology entrepreneurship course series is, to an institution. If you have one or more of those courses that can expose both trainees and faculty to the ability to start up companies, and I do want to get quickly to that. So we were founded at the close of 2007. We were located in the UAB-sponsored and associated incubator downtown, Innovation Depot. For those of you that don't know about Innovation Depot in the greater network of CTSA centers, it was a vacant Sears downtown store and distribution center in downtown Birmingham that sat vacant for more than a decade, and it was turned into an incubator for all sorts of different types of companies. We were there for 4 years in 2 wet labs that were adjoined to one another and in an office space across the hall. And then because we were growing, and we graduated out of there, and we have been in our own space in a local suburb of Birmingham, Inverness, for the last 5 years, and we just signed a 3-year lease extension. And we are now 10 years old. Now, what I should say, it was great to get started in 2007. because everybody knows what happened to the economy at the end of 2008. So if we had gotten delayed in getting started, it would not have gone well. And actually, interestingly enough, the period of 2008 to 2014 was a period, where biotechnology and technology companies were struggling. In 2015, the amount of investment dollars in biotechnology finally came back to where it was in 2007. So that tells you, how long that period was. Now, I will tell you a little bit about how we survived through that period because of our unique business model in a little bit. So if anybody, again, has any questions, you can chime in through the network that's live streaming, or please interrupt me and ask me. And so this next slide, the UAB to DBM timeline and the steps, talks about what I was doing. I was growing frustrated about the pace of our basic science being translated into patients and people. And in some degree, I had a PhD, but I didn't have an MD, and you need a little bit of both to be able to get that done, right? So I was interacting with MDs within the Cystic Fibrosis Center to try to translate our stuff into patients, and we even did some pilot nasal potential difference experiments in CF patients. It's one of the non-invasive ways to test to see whether your CF therapeutic approach is working. I wanted to do more, and I wanted to do more with human cell platforms, is what I would call them, primary cultures of human cells from normal and diseased tissue, and eventually also immortal human cell lines from those normal and diseased tissue, and use that as the key ingredient on which to screen small molecules. And so that became the genesis of the company. Now, we took the idea from scientific idea to academic core to new company through 2005, 2006. I took the classes that I now are involved in directing or lecturing in late 2006, 2007. Directed and taught at the time by the late David Anthony, who was an entrepreneur in residence, who was a venture capitalist as well, who brought this series of classes to UAB. At the end of those classes, I had a business plan for Discovery Biomed, although the business plan now for Discovery Biomed looks a lot different than it did back then, and we can talk about how that morphs and changes. In April 2007 through August 2007, I started an introduction and negotiations with the Birmingham Technology Fund, and this is where timing is incredibly important. They had just finished raising the money in this fund around the same time that we were getting started, and they were looking for companies to invest in. So that was incredibly helpful and timely. During that same period, spring and summer 2007, I had UAB to DBM transition discussions. And actually it became apparent that it was probably best for me to move from academia to this company, because I had an interesting discussion with the conflicts of interest director at the time at UAB, saying, if you tried to continue to be a faculty member while doing this company, you'll be in my office every 2 minutes. So, and you'll be doing a lot of paperwork too as well. And so you may want to just move over. And so that, and for a lot of different reasons I won't go into, I did decide to resign my tenured faculty appointment and change. And so we signed a lease for labs in an office in Innovation Depot in the middle of 2007. So there were a lot of moving parts in the middle of 2007. And during that summer, I decided to resign my tenured faculty appointment and found and launch and run Discovery Biomed. And then a funny thing happened. And so we were talking with investors at the time about investing in the company. What do you think? This is a question to everybody listening from afar and near. What do you think, how do you think the investors reacted when I told them that I had decided to resign my tenured faculty appointment? What do you think that they said?
Great, you can be full-time at the company.
That's true, and that's exactly what they said. In fact, they were so happy that I was fully invested in the thing and I wasn't going to be splitting the time, that they put in more money and they accelerated the process for that to be able to happen. And I had absolutely no idea that that was going to be the reaction that they had. So that's an example of that. Now we're going to talk later on about ways where the tenured faculty member doesn't have to leave and can still form a company, because I know every university doesn't want to have, you know, their talent leave and go to the commercial world, but there's a way of doing it so that it doesn't have to happen that way, and do something as risky as I did, because it's still, my investors are still very fond of telling me that I'm still doing this entrepreneurial risky thing while my significant other has a stable job still at the university. So she's saying I'm a little bit not, but anyway, but we can talk about that. So in August through December 2007, we got a bridge loan to begin. That was part of our initial $600,000. And I left UAB formally at the end of September, and I started at the company in the 1st of October. So never really retired, just switched on over. So in the fall of 2017, we got our first $600,000 in total of common equity investment. We launched formally on January 1, 2008. Again, timely. If we were delayed any further, it would have been kind of a difficult slog. And then in the early part of 2008, we got another $600,000 in common equity. And again, if we had been delayed, it would have been difficult. Does anybody have any questions at this point? Yeah, go ahead.
So in your timeline, October 1st, you're an LLC, but
Yes.
then you're a C Corp.
Yes.
So
Explain the difference. Right, and why that happened.
why don't you spend the money to do one and then have to spend the money to do another one back to back?
Okay. As we know, our newly elected president has a lot of LLCs, over 500 of them, okay? And we had one, okay? So LLC stands for Limited Liability Company. And one of the reasons you form one of them, especially when you're starting, is that the tax implications flow through the company to the investors that actually put money into the company, okay? As it turned out, some of the investors in the Birmingham Technology Fund is a public-private investment fund. It's not necessarily a venture capital fund, and it's not an angel investor fund. It's a little bit of a hybrid. Some of those investors could not take those flow-through tax losses. So when the money came in from the Birmingham Technology Fund, during that papering that up, we had to convert from an LLC to a C corporation because of the tax implications that happened. A C corporation pays taxes. An S corporation is slightly different. It has slightly different tax law, but it has to pay the taxes involved. Now, the nice thing about not being profitable and being break even for so long, and I'll mention that going forward, is that you're not making a lot of profit and you're not paying any tax, right? As soon as you start to make profit, then the tax man comes knocking on the door and says, okay, I want some of your money. But, and that's starting to happen now for us. But that's the reason for the change. You know, we became Discovery Biomed Incorporated when all of that legalese went through and when we formally launched on January 1, 2008. Any other questions? Okay. Questions from afar? Okay. So company highlights. Okay. So we were founded in 2007. We've been based in Birmingham, Alabama all the time. We've been operating on lean startup principles throughout. That's very important. All of our scientific equipment, with the exception of a few things, is new, because I don't like to buy used equipment because oftentimes it breaks and you don't know who had it before you had it and things like that. But all of our office furniture at Discovery Biomed is used. It came from our house. It came from our used office furniture dealership, you know, things like that. And, you know, we don't have fancy decor at Discovery Biomed. It's mainly labs and some office space and things like that. We are in our 10th year of operation. Yay! You know, surviving. I'm still being there and doing that. This is the first time I've run one of these companies, but we have some biopharma consultants, ex-biopharma consultants, and other, you know, business veterans that are advisors to us and are incredibly helpful in doing that. We had in total, over 2007 through 2009, almost $2 million, $1.95 million in seed investment from the Birmingham Technology Fund. We have not required subsequent investment since then, okay? And here's the reason why, okay? On the one hand, we've been very good at and very fortunate at getting small business innovation research grants. SBIR grants is what they're called. There's another type of grant called Small Technology Transfer Grants, STTRs. Those are for companies, and they're also very useful, for companies that are forming and still licensing the technology from the university or optioning it in to be able to use. So for companies that are forming or are still in concept, you can apply for that money, and then you can actually switch to the SBIR track if you want to after your company is formed. But in both cases, the small business concern applies for that money, but with the SCTR in collaboration with the university. And we'll have another slide that explains the differences between those, what types of grants you can get, and then a new mechanism that's popped up recently as well that I'll mention. But over the last 9 years, in the beginning of year 10, we've had 8.7 million in SBIR grant awards, and another 1.2 million is pending. We hope that a budget is passed soon, and they can figure that all out, and they'll be able to release those funds. And the other thing that's been helpful has been the fact that everything we do in humanized drug discovery, and I have another slide about that, and that includes creating the primary cultures and immortal human cells on which we create platforms for screening, humanized drug discovery or screening on those human cell platforms, and something we call humanized lead therapeutic asset profiling, which is a little bit different. Let me explain that. If another industry client or academic client has already done the screening, has already done the high throughput screening, and already has their own therapeutic assets, biologics, small molecules, antibody drug conjugates, you know, whatever they have, if they already have that, we're of value because they come to us and they say, okay, tell us which of these in your panel hits the target, the phenotype, or the pathway in relevant human cells, both diseased and normal. And if we can tell them which one or which subset of those works really well in normal and diseased human cells, that's a critical box to check for them, because that tells them which ones they can take into preclinical testing and ultimately into clinical trials in the FDA. Saves them time, saves them resources, saves them money, the number one important thing, right? And saves them also the number of animals that you use in those studies too, which is another issue. So let me say it this way. Those three numbers, 1.95 million, that was the money that helped us start up from scratch and set up the initial infrastructure that we've added to as we've gone forward. The 8.7 million, with a little bit more pending, has helped us fund our own R&D programs where we've been discovering our own drugs for particular disease indications at Discovery Biomed. And the 3.5 million and growing in CRO services revenue is where we've done exactly the same science with exactly the same infrastructure as a service, initially for academia and eventually for industry. Yes?
So at this point, when you are doing CRO work, who is doing all your conflict of interest, like, you know, like, because you are getting somebody's compounds?
Yes.
And selling the agreement, so companies do it, or you have your own people kind of taking care of some of this?
It's the same team of people that do both. It's the same science and the same team. Do you want me to repeat the question? Sure. The question was, how do you handle both the CRO services work and the compounds that are coming in that you screen, and while you're doing your R&D work? And number one, it's tricky. But number two, the only way we can do that is by having the same people that do the same type of experiments for R&D that we do for CRO services. Now, there is an issue of time, and so you have to do time management of that so that you can separate the services work from the R&D work and get both accomplished. And we're at a point now where we're actually adding new people into the mix to be able to handle all of that, because we have multiple large company CRO services clients right now that we're doing programs for, none of which I can name, unfortunately, because we're under confidentiality agreements with all of them. And we're continuing our R&D work. Now, the one way we can handle that is that a lot of our R&D work now has gone beyond us, and they're actually other CROs, contract research organizations is what they stand for, that are doing work for us for our R&D program. So we're actually not physically doing that work. So we have time and infrastructure to be able to do the CRO services project. So it's a juggling act. It's a managing act. We now have so much CRO services work that we've had to add people. So I've added a person to help me with business development. I've added a person to help me with financial and legal issues that are involved in all that, the papering up of all those things, and also multiple people to help with the science and the data management and the data analysis and things like that. So the CRO services revenue, albeit not a lot over a 10-year period, that has covered the overhead for our company, okay? The bottom line in all this is that all three of those numbers added up on that slide in the company highlights has been absolutely necessary for us to survive. And if we didn't have each one of them, I wouldn't be here talking to you today. I'd be doing something else, okay? Because that's been absolutely necessary for us to survive. And so in some cases, most of our monies are coming in from SBIR grant awards. In other cases, they're coming in from the CRO services business. This year is probably the first year where it's going to be a 50-50 even split. That's the projection right now for the financials. And actually, on average, so for the last 6 years, 6.5 years, since end of 2009, we've been absolutely break even with 1.1 million revenue average over that period of time. 2016 revenues were nearly 1.7 million, and now this year with this uptick in CRO services business, catching up with and pulling even with our R&D work that we're doing, we're projected to meet or exceed $2 million in revenue, which is, it was hard to get to 1 million. It's been even harder to get to 2, but you know, we're working on it. And it's been the dual business model of internal humanized drug discovery R&D and customized humanized drug discovery services that has kept us alive. And the only way you can do that is with a common team, a common infrastructure, common expertise, common approaches and methods, common principle, and things like that. And that core philosophy, the use of human cells, normal disease at all stages of drug discovery, has not changed. That has been set in stone from the very beginning. Our business model has been tweaked and moved and pivoted and so on and so forth based on, you know, percentage of services versus R&D work, but that core philosophy has not changed. And we've got a newly redesigned website if you want to go see it. We've added some new personnel, so the personnel page will change soon, but that's kind of the same. Any other questions at this point? Okay. What's that? Our patents are handled by a law firm in Atlanta, and actually the person that runs that shop was a PhD that trained here at UAB, actually, a while back. And now she's in Atlanta and she runs a great IP firm. And so we have patents for 2 of our 3 R&D programs, and for the 3rd one, we have not, actually we're holding our small molecules as a trade secret and we haven't patented them yet. But as soon as we know which ones we're going to progress, we're going to patent by chemical series, actually, because it's easier to do that. We've had previous patents where we had multiple chemical matter in the patent, and the patent examiners make you break that up. So we're patenting by chemical class or chemical series that we found, and we've learned that by doing as well, because that's a whole another sector that, you know, I've learned by doing it. It's quite complicated and quite interesting at the same time. So this is a schematic of what we do, and I've kind of already covered it, but we implement disease-relevant human cells at all points of the critical path. So from generation of the primary cultures from human tissue, to immortalization of the cells to expand them enough to be able to do high-throughput screening, all the way through primary HTS or high-throughput screening, through hit and hit-to-lead validation, to lead prioritization and progression. So academia and small biotech will adopt using intact living human cells as the key ingredient in our platform at the earliest points, at time 0. Biopharma, you can't quite go in and tell them you're doing it wrong, you need to do it this way, because they'll show you the door. But what you can tell them is, you know, when you have your hit the lead or lead compounds, you know, come to us and we can do some profiling in the relevant human cell platforms and tell you which drugs you might want to focus on, you know, going forward. And I would argue that this approach can be applicable to any disease. It adds value to the drug discovery critical path, The humanization of drug discovery increases disease relevance and decreases risk, okay? If you have the HTS assay well designed and you have some critical validation steps so you can identify true positives and true negatives to what you're doing, it can lower the attrition of those attractive, interesting compounds going forward. Any other questions at this point?
Eric, could you tell us a little bit about the platforms? Because you talked about immortalized
Sure.
human cells, but I only know about immortalized B cells because I'm
Yes.
an immunologist.
Right.
So I wouldn't be surprised if the only relevant cell was a B cell, but you might have a somewhat broader view. Could you tell us a little bit about how
somebody asked us to do that recently, and it's not easy because those are not adherent cells. So, so the way you do the assay is so tricky because those cells are in suspension. So the question is, tell us about some of the human cell platforms you're using and how. So I'll give you 3 examples and actually perfect segue to this slide. So our internal drug discovery has focused on 3 platforms going forward. Now, we've done a lot of other stuff, and tried to get SBIR funding, but these are the 3 that have kind of caught fire and gone forward. So for the respiratory CFTR ENaC program, we actually selected an immortal CF human bronchial epithelial cell line that was very adherent, that's very important for high throughput screening experiments, and was homozygous genetically for the most common mutation in cystic fibrosis, the Delta F508 mutation. And we used it unmanipulated to do screening of small molecules. And what I mean by that is we didn't overexpress the mutant form of the protein. We didn't overexpress any reporters. We didn't overexpress anything. We just took that CF human bronchial epithelial cell line and screened small molecules, looking for small molecules that rescued lost halide membrane permeability. Okay? Now that's not completely targeted to Delta F508 CFTR. That could find things that upregulate alternative populations of chloride channels. And actually one of the hit delete classes we have does not touch Delta F508 CFTR. It upregulates other chloride channels. And we're trying to investigate which ones. We do have a class that we have moved forward to be our lead class. Out of that screening, we think we found it because we did the screen that way with that relevant human cell. It corrects Delta F508. It also potentiates wild-type CFTR, and by virtue of what it does to CFTR, it activates its ability to be a signal transduction molecule in the cell, and by virtue of upregulating CFTR, it also inhibits ENaC, which is the other dysregulated sodium channel in cystic fibrosis. So we are now in due diligence with the biopharma right now that is doing their own internal experiments on the lead molecule right now to determine whether they're interested in doing a partnership deal with us. And they're actually more interested in the fact that this is a novel type of ENaC inhibitor than they are that this is a CFTR ligand. So that was done on a CF human bronchial epithelial cell line. What we're doing in autosomal dominant polycystic kidney disease is we're making ADPKD primary cultures that are unique and normal human kidney primary cultures that are unique, and creating platforms on which to do discovery, not only for ourselves, but also for clients who are very interested in doing that right now, especially in industry, none of which I can name. So the way we're doing that is we're growing these cells in 2D and in 3D biogel platforms to be able to do this type of analysis. And I was talking with Molly before about the fact that we can isolate primary cultures, 24 different primary cultures from a cystic kidney, and we can put them in biogels, and some of them make cysts and some of them don't, and for the life of us we don't know why, and we're trying to figure that out. But that's an example of the relevant cell platforms we're working on right now, and we do have a lab to marketplace SBIR which is funding our platform creation for that particular disease, and we have an anti-proliferative ligand that we're profiling in those platforms right now. And in the context of making ADPKD and normal human kidney platforms, we also have diabetic ADPKD and diabetic kidney because some of those donors happen to be diabetic. And so And so we're continuing to process donor tissues that come in. I should say for everybody out there saying, okay, you're processing human tissues, how are you doing that? The ADPKD kidneys are often removed before you lose renal function because they get so large that it's the intense flank pain in the abdomen that necessitates their removal, and it's often done before dialysis or transplantation. Normal human kidneys that we get are normal in quotation marks. They're not viable for transplantation. They come to us. In some cases, we can process them into primary culture. In some cases, we cannot because of the disease complication that's come up with it. For example, we had somebody with a kidney that came to us that had profound hyperlipidemia, triglyceridemia, and there was so much lipid in the kidney that it killed every cell we tried to put into primary culture. So I should say that none of these normal kidneys that we get are viable for the transplantation chain. And then the final one that we've done some work on is that we take human pre-adipocytes from normal diabetic, obese, and diabetic obese individuals, and we differentiate them in culture to be differentiated human adipocytes. And anybody that's familiar with fat cells, a pre-adipocyte looks like a lot of other cells that you see in culture. A differentiated one accumulates all these lipid granules in it, so it has little balls of fat, hundreds of little balls of fat in each cell. And it's at that stage that we do studies. And we were looking for small molecules that upregulated the endogenous production and secretion of adiponectin, which is a good adipokine that is present in all of us in quite abundance in the plasma, that is lost as diabetes and obesity progress. And we have cultures of adipocytes from diabetic obese donors which generate about 5 to 10% of the adiponectin that you would see out of normal adipocytes. And so we are trying to find small molecules that will upregulate the endogenous production and secretion of adiponectin. There was a biopharma that tried to make an adiponectin biologic for 10 years. And it's such a complex multimeric glycosylated protein that they failed in making it. But it's an interesting program, and there are multiple biopharmas interested in that program. We just have a few more boxes to check so we get there. So those are 3 examples. Another thing we're doing right now is we're actually getting primary cultures of human taste bud epithelial cells, and we're immortalizing them now so we can do taste receptor ligand discovery for an academic client that has several third-party industry clients that are interested in doing that taste receptor ligand discovery. So that's very interesting. I can't name any of those large third-party clients either that are working with our academic center collaborator. But basically, now we focus mainly on renal, urologic, respiratory, and metabolic therapeutic spaces, both for our R&D and for our CRO services, just because we need to have some focus. I'm one that tends to be defocused, and all my people on the team tend to try to refocus me because we do other things. But we've done other cell platforms like human cardiac myocytes. We tried to do a pilot program. We tried to do brown fat-derived adipocytes at one point. We tried to do islet cells from the pancreas at one point, although that was incredibly difficult to do. So we will go out of those therapeutic areas, basically if the money's good, if you will, and if somebody wants to do that. So, and one of those studies, actually, pilot platforms right now that we're doing is in urine and human reporter cell lines, one of which is THP1 cells, which is a thymocyte that is used in immunology right now. You have another question?
Yeah. So what's the source of your small molecules?
Excellent question. So we have a library of small molecules, that's the question, that comes from 5 different commercial vendors. So because of that, it has some diversity to it when we do a full screen of that 70 to 75,000 compounds. In biopharma terms, that's not a lot of small molecules. I mean, they like to screen 1 million compounds in the least unit time, and get the data as fast as they can. But from that library, we found meaningful stuff, and in all 3 of their programs. Now, a services client may want us to screen their own library, so they may send it to us, or they may send us a targeted collection of compounds for us to screen, or they may send us that hit the lead or lead panel for us to screen. So in that case, we're screening those chemical entities that they send to us. And oftentimes, I should say, those are coded and blinded. We don't know what they look like. And that helps protect their IP because, we don't get a look at that. So we screen the coded and blinded compounds, we give them the data, and they unblind it at their end, oftentimes.
Many of these compounds are already patented, right, by the companies.
Well, before we go to the expense of doing that, we take a look, with our IP attorney to make sure there isn't any, chemical composition of matter out there that matches with what we have. And so far, for those that we progressed, we haven't run into that entanglement yet. But that's an excellent question. That's it. How do you know if something you found has already been found by somebody else, and you take pains to make sure that that's not the case?
So just a gentle intrusion here. It's 5:45, and just
sure.
to be cognizant of people's time.
Sure.
If there are 5 to 7 minutes.
Sure.
To make sure that we cover the
yes,
things that you
the
think
new
are
company creation
key secrets.
slide. Yes. So let's do that. So the next slide is a slide that we talk about a lot in the idea to IPO class, and I did want to touch upon this, and there's several different ways to do this, and I have to say that discussions are happening with UAB leadership and tech transfer about how better to do this, and everything's going in a positive direction. I was asked to be part of an external committee that looks at this about better ways to be able to do this in biotechnology, and so this is all happening in a good way. There's a talk about a faculty accelerator that actually is going to happen on campus. They'll be able to do that. All of those things are valuable vehicles to think about at the partner institutions as well, to get that going, to pilot it, to find out what the best practices are to begin with, and then to build upon that. It's important. And so I've shown this slide at the New York Academy of Sciences, West Virginia University, and also at UAB. So when I'm telling this to a class that's mostly trainees, PhD students, postdoctoral scholars, and even some faculty that are interested in taking this class, I say that the university academic inventors are like fully differentiated cells, okay? All right? They're fully differentiated. They like where they are. They have tenure. They're not going to be often wanting to give that up easily, okay? But they don't have to. That's what I'm saying. They don't have to do what I did to be able to make this work, and this is my point. So they can stay on the basic science side. We have lots of PhD candidates and postdoctoral scholars that are emerging from here and many other, fine institutions. They are less differentiated cells. There's still an ability to turn them down different career paths. So why not have those trainees populate the new company on the other side? And then on both sides of the well of valuable, untapped university technology, it has Blaze the Dragon here for UAB, but it can be any university in the network, okay? The newly minted trainees, PhD students, postdoctoral scholars, a combination of the two, can form and populate the new company on the applied science side of the well, while the tenured academic inventor that trained these people can continue to innovate on the basic science side, because there still are things to do on that basic science. There are still things to be learned, nuances to work on. Why not apply for the SBIRs and the STTRs and the CRP grants? There's a future slide that talks about that, together, and work on that together. So the faculty inventor can consult with the company. They might even be able to do a sabbatical or a short leave of absence to help form it in the beginning, but go back to the basic science side while the trainees do the applied science side. They apply for a grant, and actually the company gets that money, but they can subcontract that money back to the academic inventor. So there is a way for tenured academic faculty to access SBIR, STTR, CRP dollars that are designed mainly for small business concerns, okay? And that is a way to find homes for PhD candidates and postdoctoral scholars that are looking for homes, and it's a way to do both applied science and basic science together. I see it as covering both applied science and basic science. I don't see it as a dark side or a light side. I guess that's because I spent 11 years as an academic investigator and 10 years now as a biotechnology company person. So equal time in both places now almost. So I see it as being a symbiotic relationship if, you can find the best approaches to be able to do that. And so the applied science side eventually can find investor funding in whatever forms going forward to help fund it, and even some of that money can find its way back to the basic science side. And there are things to do to work together. You know, for example, we're doing drug discovery on the applied science side, okay, but we can't do all the experiments. So in some cases, we've asked UAB faculty or faculty at other institutions to do key experiments for us that we can't do. That's in the form of a subcontract out of us that goes back to the university to do that. So there are multiple ways for a university to benefit.
We got a question here online.
Sure.
It says, do undifferentiated scholars have sufficient CVs to serve as lead investigators of SBI office?
Bam, excellent question, okay. We have run into that where my younger investigators have applied as solo principal investigators on SBIRs, and they get the comment back.
Muted. Unmuted.
Okay, so that's the way we mitigate that. The same way we can mitigate that in this case on this slide is that the academic investigator and the new investigator that's on the company side can go in as dual PIs. That is permissible. I see that a lot in applications that come into these panels where they've had this issue where new investigators are trying to get their first grant but don't have that track record of NIH funding. Well, the only way you can get that is apply for grants, right? So that's an excellent question and a good question. And let me speed through a few. Oh, and it's right here on the next slide because maybe I thought we needed to move this up. So on several subsequent slides that we probably won't get to, I talk about different key principles about startup companies, what to watch out for, and different business models of how you can get started. You don't have to set up from scratch like we did. You can actually set up and piggyback on existing companies that are already doing it until you get legs under yourself to do it yourself, okay? We have several companies that are piggybacking on us now, or will in the very near future, out at our facility. And one of my board members has started a new company with technology that he licensed out of Vanderbilt. He's right next door to us. So between the two of us, we have 9,000 square feet, and we don't use it all the time, and we don't have it all populated yet. So other companies can piggyback on us to do that in a kind of an accelerator-like complex. But let me cover this because it's very important, and we can ask questions at the end. So an SBIR grant, Small Business Innovation Research Grant, submitted and largely driven by a small business concern with 500 and fewer employees where the principal investigator is a key member of the company that devotes 51% or more effort to the company, okay? It can be a private company or, wait for it, it can be a public company. I'm not kidding, okay? We had this issue of a company that was private and was doing an IPO and had grants in, and we were like, what happens if they IPO? Can they accept that money? And the answer is yes, as long as they are less than 500 employees and they have somebody that's 51%. Who knew? I had no idea, right? We had to ask a lot of people about that before we got an answer. But anyway, that's the case. STTR grants, okay, submitted by a more newly forming small business concern in collaboration with the university institution that it's being derived from. Same size company and percent effort rules, but it usually applies to a company that's in an earlier stage where the term sheet and the licensing agreement are being ironed out between those inventors and the university. And then there's a new one, and I've actually emailed into the SBIR, STTR director himself to continue to support this program. For companies that hold phase 2 or phase 2B SBIR or STTR funding, you can apply for a commercialization readiness pilot program grant, and that is a less restricted grant where you can use the bulk of those studies to check a key box on the way to getting an IND, especially to do something important in IND enabling studies to be able to do that. So for a phase 1, the recommended level of funding is $225,000, and it's for 6 up to 12 months, depending on how much time you need, okay? You can go over that level with appropriate budget topic justification, okay? And if anybody is interested in knowing that, there's a way to do that, and I can tell you how to do that. For phase 2, once you progress to phase 2, you can get 1.5 million, the recommended level, or a little bit more with appropriate justification, and that's for a 2-year period, okay? Probably not for a newly forming small business concern, but for somebody that's actually done these SBIRs for a while, you can actually apply for the whole thing in what's called a phase 1, phase 2 fast track SBIR. We tried when we were a new company, not a good idea. We needed to get a few years and a track record of SBIR funding before we went and did one of those, because you're asking for more money over a long period of time, and they're reviewed with a little bit more scrutiny, actually. And then the phase 2B is a $3 million award over 3 years. You can only apply for one if you have a phase 2 SBIR, and that usually has some strings attached to it where you need to go get partial or full match from an investor group or a biopharma that is funding this in parallel along with the phase 2B grant. And the CRP grant I just mentioned, it comes in 2 different forms, and only some of the institutes are participating in it, and it's called a pilot program, and it is a pilot funding mechanism right now. They haven't decided whether they're going to continue it or not. Okay. And the important note again at the bottom of this is inventors that stay within the university can still derive some significant monies from both types of these grants by forming a company on the applied science side, getting their trainees to populate it, and by working them together. And that maximizes value for the University of Origin. They've spun out a company, but the university is also indirectly getting some of that money back if it can be constructed properly and things like that. So are we right at the time? Should we ask for a question?
Do we have questions?
Questions from here, questions from the network. I'll give you these slides and you can post them and stuff like that. I think.
So one of the questions I would have is when you're applying for a phase 1,
Yes.
how mature does both the business concept and the underlying science need to be? For many of us in the room, we may understand the vernacular of the NIH space quite well.
Right.
But translating that experience into the language of an SBIR doesn't necessarily come naturally.
In form and in length and in approach, a phase 1 SBIR or STTR is a lot like an R21, okay? You technically don't have to have preliminary data to be able to apply for one. But as a reviewer of these, I would strongly encourage you to have some, okay? And once you open that Pandora's box of putting in preliminary data, it's got to be pretty solid, because then the reviewers start to go look at it and scrutinize it and things like that. I'll give you an example. So it's very hard to go in cold and get money for a drug discovery program if you don't have any preliminary data, because it's a huge leap of faith for reviewers to say, oh, yeah, they know what they're doing. They're going to find something. So, so what we do is we have the human cell platform created. We have bioassay optimization done on it, and we do a pilot screen of a couple of thousand small molecules, and we do even a couple of validation steps so that we can show them a concentration response relationship for an activator or an inhibitor that's come out of that screen. If we have that pilot data package embedded within a phase 1 SBIR, that increases our chances tremendously, okay? So a phase 1 SBIR or STTR grant is a lot like an R21. They're 6 pages long, not a lot of space to get your point across, especially if you have preliminary data. So I use some of the other sections in there that are less page length constrained to make key points, about what we're doing and who's involved and things like that to be able to do that. A phase 2 or a phase 1, phase 2 fast track has to have pretty nice, compelling preliminary data in it to get it over the hump. Now let me say this about scoring of these, okay? None of these grants are percentiled like our grants, okay? They are only, funding decisions are only made based on priority score, okay? One thing that may not be readily apparent is that over the last 5 years, was a reauthorization of this program where each year 0.1% more was added to both the SBI or STTR program mandated by Congress for every institute for this program. It's actually taking money away from the rest of the pie. It has been reauthorized from 2017 through 2022. I don't know what that looks like. I think they're probably trying to maintain the same funding levels. And it's actually a rider on the Defense Act, and it should be signed, but it hasn't been signed yet by the new administration. So because of that added money outlay from every institute, which is now all told about 4.5% of every institute or center's extramural budget, the priority scores that are needed to get funding are in the 30s, okay? All right? And usually the not discussed line for a score when people are sending in their preliminary scores is around 40. So if you can get discussed, there's a pretty good chance that you're going to get funded on the original submission or more likely on the revised submission and get these grants. The odds are much better to get these. And so universities are trying to find, clever ways to be able to access more of that funding.
So to respond to,
Bob's question really, the key here, which I think you and I talked a little bit about, is to find the right partnering company to put the science idea to apply for even phase 1.
Right. So you could partner with an existing company to be able to gain access to these, or you could form your own company, to be able to do that on the applied science side, or you could partner with an existing company to get started. You could piggyback on them, and then when you got your own legs and your own funds, you could find your own lab and space if you wanted to do that. And that's the idea. That's the accelerator idea, is that you're in a space where you're sharing lab and office space, and you're getting legs, and you're applying for these, and you're trying to get funding. And then when you have sufficient funding, SBIR, STTR funding often begats investor funding later on because you've been able to win these awards, and then they don't have to do their own due diligence on the science. The reviewers have done it for them, so they're more apt to follow that money with their own money afterwards. Yeah. So.
I have a question.
Sure.
This is Nancy Rush. I'm the chair of pharmacology. I'm talking to you from UAMS here.
Okay.
But can you just comment on your personnel structure and how you expanded the company and got business expertise?
Excellent. So actually, before I left, I had somebody who is a co-founder down there setting up the company. So the first employee was not me. It was actually somebody that was setting up our bank accounts and our financial structure and getting QuickBooks and ordering equipment and office equipment and lab equipment and help getting that started. So it initially started as 2 of us. I rapidly understood that I needed another biologist and a chemist to help with the compound libraries because we needed somebody to manage all of the molecular libraries we had and be able to organize all that and things like that. So in that first year, we started out with 2, and we ended up with 4 full-time employees. We had a couple of part-time interns that came down and joined us. At the time we left Innovation Depot, we had 6 full-time employees and a few part-time employees and a few biopharma consultants, and we have maintained that level of 8 to 10 full-time and part-time employees until now. And last year we had 4 full-time employees and 14 part-time interns, many of whom were derived from a couple of different UAB programs, master's students, PhD students, even a postdoc or 2 that came down and spent a period of time with us. So it was 4 FTEs and 14 PTs, and this year it'll be 6 full-time employees and 12 part-time employees. So we have 6, and those 6 full-time employees, I should say, how do you accomplish that with 6 full-time employees? Those people wear multiple hats, okay? So, and they're all PhD holding people who are self-motivated, can manage their own scientific projects while also handling other types of roles within the company. So those people have to know how to take off one hat and put on one hat and go back and forth and whatever. They all have hat racks with many different hats on them. So and that's how you can do it while keeping overhead down. So.
Are there, excuse me, are there other questions that might be from UAMS or Utah coming in?
I'm glad that Utah's on the line. Utah is very vibrant in terms of an entrepreneurial, ecosystem. So they're a nice model to look at. They've got a lot of different interesting startups, and they've got an incubator there as well, where they have a lot of interesting companies. So.
Questions
from the UAMS site, we really enjoyed your talk.
Oh, thank you.
So I think it was really welcome. We're kind of at the point where we have a lot of startups that are thinking of starting and a lot of students involved, so we really enjoyed it.
Good, excellent. Glad to hear it.
Well, we're also very grateful here from the UAB side as well. So big round of applause for Eric.